In today's video, we're going to be moving on and looking at topic 1B for AQA A Level Biology. So this series of videos is going to cover DNA and RNA, which are what we're looking at today. We're also going to be looking at inorganic ions, ATP and water, which will be topics I cover in subsequent videos. So let's have a look at DNA and RNA. These two molecules are what is known as nucleic acids, and they are found in every living cell, in every organism, and their role is to carry genetic information. So in humans, every single cell that we have, except for red blood cells, contains a copy of DNA. And DNA stores our genetic information, so it can provide instructions for when we need to grow and develop. So whether it's a child who's going through a rapid stage of growth, or whether it's an older person whose cells are replacing due to maybe damage or just old age, they will contain a store of DNA. And DNA is basically there to give instructions to make proteins. So every protein that we need in the body is encoded by DNA. And so DNA really is there to help us code for the molecules that we need in order to live. Now, as I said just a second ago, red blood cells don't contain DNA, and this will be something that we cover in a later module, but this is essentially just because they need to maximise their space they have for oxygen transport, so they don't contain DNA. And if we look at RNA structure is very similar to DNA, but its function is to transfer genetic information from DNA to a molecule known as a ribosome. And ribosomes are often described as protein factories because essentially they act to make proteins, and so ribosomes will read the RNA that is transported to it, and it will make polypeptides by a process called translation. And translation we will have a look at in topic 4. But as we know, polypeptides are the primary sequence needed to make proteins. These polypeptides will then go on and they will fold and they will make the protein that is encoded for by this RNA sequence. So DNA and RNA are made up of small molecules called nucleotides. And if we have a look at the basic structure of a nucleotide, they contain a pentose sugar, and what that means, pent meaning 5, like pentagon, and that just means that there are 5 carbons within this sugar. So they all contain a pentose sugar, and attached to this sugar is a phosphate group and what's known as a nitrogenous base. Now, this nitrogenous base is an organic base, which basically means it contains carbon, but it will also contain nitrogen. So now we're going to move on and talk about ribose and deoxyribose sugars. And these are the types of sugars that are present in DNA and RNA and accounts for some of the differences between the molecules. So if we have a look at DNA first, we can see that we've got our pentose sugar, our phosphate group, and our nitrogenous base. However, the sugar in DNA is known as a deoxyribose sugar, and this can be seen in DNA's full name. It's known as deoxyribose nucleic acid, so DNA. And DNA nucleotides will all have this same deoxyribose sugar, and it will all have the same phosphate group. However, the nitrogenous bases can be different, so they can either be adenine, cytosine, guanine, or thymine. You don't need to remember these names, but you do need to remember that the bases are A, C, G, and T. So in RNA, the sugar instead is what's known as a ribose sugar. Again, in the name, ribose nucleic acid, RNA. And RNAs also, as well as their sugar, contain a phosphate group and a nitrogenous base, but the bases in RNA are known as adenine, cytosine, guanine, and uracil. So they've got the same bases as DNA, however the thymine that we'd find in DNA, in RNA is replaced with the base uracil. So as we said, DNA and RNA are made up of these small nucleotide molecules. And when we make a full strand of DNA or RNA, I form what's known as a polynucleotide. And a polynucleotide are polymers made up of nucleotides. So these nucleotides join by a condensation reaction, which is something we should be very familiar with now from topic 1. And this condensation reaction occurs between the sugar on one nucleotide and the phosphate group on another. And I'll be able to show this much easier on the diagram on the next slide. And when this condensation reaction occurs, it forms what's known as a phosphodiester bond. And again, the name gives away what this bond consists of. So it essentially consists of a phosphate group, phospho, and 2 ester bonds, diester, phosphodiester bond. And we can demonstrate this. So here would be our pentose sugar. Here's our phosphate group, and the bond between this phosphate group on the left-hand side and our pentose sugar is 1 ester bond, and then at the bottom left-hand side of the pentose sugar, that carbon would react with the phosphate on another nucleotide, and this would form our 2nd ester bond. And obviously our nitrogenous base is also present. And these bases, although they're not involved in the formation of phosphodiester bonds and forming what's known as the sugar-phosphate backbone, these bases will stick out and they will pair by something called complementary base pairing, which we'll move on to in just a second. So as I just said, as these sugars and phosphates bond and form these phosphodiester bonds, we get what's known as a sugar-phosphate backbone, which is just a long strand of phosphate, sugar, phosphate, sugar, phosphate, sugar, so on and so on. So when we have a look at the structure of DNA, we can see this on the right-hand side of the screen. If we look at the left-hand side, we can see we've got these purple pentagons with a letter D in the middle, which represents our deoxyribose sugar. We can see that in between the sugars, we've got these nice yellow circles with the letter P in that represent our phosphate groups. And we can see the sugar-phosphate backbone. We can see we've got a sugar bonded to a phosphate, bonded to a sugar, bonded to a phosphate, and so on and so on. And as we can see, our bases are sticking in to the middle. And these bases, the bases from one strand will interact and bond with the bases from another strand. And this will form our DNA double strand. And they form hydrogen bonds by a process called complementary base pairing. So adenine always bonds with thymine, and cytosine always bonds with guanine. And adenine and thymine, or A and T, will form 2 hydrogen bonds between them, and cytosine and guanine, C and G, will form 3 hydrogen bonds between them. So cytosine and guanine form slightly more stable structures because there's more bonds between them, and they also require a little bit more energy to break because there's 1 extra hydrogen bond. Now DNA strands run anti-parallel, which means they run in opposite directions. And we can see this if we look at the left-hand side of the diagram. We can see at the top the DNA strand is labelled as the 5' end, and at the bottom it's labelled as the 3' end. If we move over and look at the right-hand side, we can see at the top we've got the 3' end, and at the bottom we've got the 5' end. And they run opposite to each other. We'll cover this in our next video when we look at DNA replication, but this allows the enzymes to run in one direction, and it makes the process of replication much more efficient. So when all of our base pairs have paired and formed hydrogen bonds, we've got our 2 strands, and these will then twist round to give DNA its typical double helix structure. So we can see on the right, we've got this very familiar looking structure of DNA's double helix. Our sugar-phosphate backbone is represented by these yellow twisting strands, and we can see in the middle, we've got our bases represented by these little pastel coloured squares. So when DNA was first discovered or found or observed in the 1800s, scientists doubted it was the molecule that would carry the genetic code because its chemical composition is quite simple. And a lot of scientists argued that proteins carry genetic information because there's a lot more chemical variation in proteins. But in 1953, there were several experiments that proved that it was actually DNA that carried the genetic code. And around this time, Watson and Crick discovered and published the structure for DNA, which obviously we now know is the double helix. And at first they theorised that there was perhaps a triple helix, which obviously we know now is an incorrect theory, but a lot of their work was heavily based on something called image 51, which was actually taken by an English chemist named Rosalind Franklin, and she was an X-ray crystallographer, and she produced an image which clearly showed evidence towards a DNA double helix, and this was then used by Watson and Crick to develop their theory of the DNA double helix. And not many people actually know about Rosalind Franklin and her work, and the reason was Watson and Crick were awarded the Nobel Prize, and Franklin was never actually awarded the Nobel Prize for her contribution to this work because she had died 4 years before the Nobel Prize was given out, and I'm not sure if it's the same, but at the same now. But at the time, they did not award posthumous awards for the Nobel Prize. So her work is actually largely overlooked when we talk about the structure and discovery of DNA structure. So going off on a bit of a tangent there, but it's quite an interesting little factoid. And if we think about the structure of RNA now, again, this will be covered much more when we look at transcription and translation, but RNA is a much shorter polynucleotide chain than DNA, so DNA can be made up of tens of thousands of nucleotides all bonded to make this very long sugar phosphate backbone. RNA will be much shorter, and RNA is actually also single-stranded, so its bases don't complementary base pair, and it just forms one single strand. So let's recap what we've covered today. We've learnt that DNA and RNA are used in storage and transfer of genetic information. We've learnt that they are made up of nucleotides, which consist of a pentose sugar, so deoxyribose sugar in DNA or ribose sugar in RNA, a nitrogenous base, so in DNA that's adenine, guanine, cytosine and thymine, and in RNA that's adenine, cytosine, guanine and uracil, and they also contain phosphate groups. We know that polynucleotides form via a condensation reaction, and these are joined by a phosphodiester bond, which are two ester bonds from the phosphate groups to the pentose sugar. We know that A and T, or adenine and thymine, are complementary base pairs, and they form two hydrogen bonds between them, and cytosine and guanine, or C and G, are complementary base pairs, and they form three hydrogen bonds between themselves. So now that we've covered this information, it's important to go back and review what we've learned and what we've covered, and then complete the following test questions. Number 1, describe the structural differences between DNA and RNA. Refer to the number of strands and the bases. What are the names of the pentose sugars in both DNA and RNA? Draw the bonding that occurs in a polynucleotide strand. And the fourth one, complete the following graph. So we can see on our y-axis we've got the percentage of bases in a DNA sample, and on our x-axis we've got A, C, G and T, and you can see that we've got information for the percentage of bases in a DNA sample for adenine and guanine, but you need to complete the graph and draw in the bars that represent the percentage of cytosine bases and thymine bases in the sample. That's all for today. Thank you for listening.